This is the first part of the case study. I promised you that I would do video versions of the two lectures just because we had a little push for time. Okay, the topic is targeting histone methyltransferase. This is a class of enzymes. The specific enzyme we're going to look at is called LSD1, which stands for lysine specific demethylase 1, which is particularly implicated in cancer therapy. First of all, we need some background, and in fact we need a pretty extensive background to follow what's going on. So I'm going to introduce epigenetics to you, which you may have come across with Jeremy Mills anyway. The term epigenetics is defined as heritable changes in gene expression that are not due to alterations of the DNA sequence. In recent years it's become more and more evident that dysregulated epigenetic regulatory processes have a central role in cancer onset and progression. In contrast to DNA mutations, epigenetic modifications are reversible and hence suitable for pharmacological interventions. Reversible histone methylation is an important process within epigenetic regulation, and investigation of the role in cancer has led to the identification of lysine methyltransferases and demethylases as promising targets for new anti-cancer drugs. And we're going to be doing enough background to put this into context. First of all, let's discuss epigenome, the epigenome. So here is a diagram of at chromosome level of the chromosome unwinding. We'll talk about this fragment in a moment. Then you start to recognize what's called supercoiling, which is now uncoiling into effectively the DNA ladder with base pairing here. Now, two main components of the epigenetic code are DNA methylation. These are methyl marks added to certain DNA base pairs, and generally the methylation represses gene activity and shuts or silences genes so that they don't get expressed into their proteins. Histone modification, and at this level, and this is the area we're going to talk about in the next few slides, histones are required to condense or wrap up the superhelix into the chromosome, the compact chromosome within a cell. A combination of different molecules can attach to the tails of strongly basic proteins called histones, and I'll explain why they're basic in a moment, and these can alter the activity of the DNA wrapped around them. So let's take a closer look at the histones. Don't get too frightened by this diagram. It is, however, to show you that there is a level of complexity of post-translational modification, and these are the varying post-translational modifications that can occur: methylation, acetylation, citrullination, and phosphorylation. And these are the tails, the N-terminal tails of histone H3 and H4 being slightly expanded, so you can see what's happening there. And you can see that what these are is these are a list of the enzymes that modify these particular residues. And you see, if you look carefully and remember from cells to systems, the residues that are being modified are basic. So K stands for lysine, arginine. You can see that the modifications occur here generally, and then here on the other tail, lysine, lysine, and so on. Histone H3, if it's modified at residue 9 specifically, and you can see that residue 9 has considerable, so here we go, 1, 2, 3, 4, 5, 6, 7, 8, 9. And we can see that residue 9 has a variety of enzymes that can modify the lysine, and histone 3H4 K4. There's a variety of enzymes that impact on this particular residue. H3 stands for histone, K stands for lysine, and the number 9 or 4 refers to the residue involved. This is a much more simplified diagram, and highlighting just the H3 residues K4 and K9. This is what's called a histone octamer, 1, 2, 3, 4, 5, 6, 7, 8. And this bundle of histones is what's helping to wind the DNA up around into a chromosome. Histone modifications that we're talking about in the last couple of slides can be written. Molecular marks may be added enzymatically by post-translational modification, or PTMs, to residues on the histone tail, in particular H3. If they're written, then they're written for a reason, and that's because they can be read by proteins involved in gene modulation. And also, epigenetic signals can be taken away again, so they need to be erased. So signals may be removed enzymatically once the gene process is no longer required. And if you think about it, this is the normal state for most genes, that they are not turned on all the time. They are expressed when that protein is required. So there has to be a process of modulation. Key signals include acetyl signals written by histone acetyltransferases, methylation signals written by histone methyltransferases. They're read by acetyl tag. The acetyl tags are read by bromodomain containing proteins. So these are proteins that contain a particular signature sequence in their protein structure that recognizes acetyl modified histone tails. Methyl tags are read by chromodomain containing proteins, and they've got a specific domain different to the acetyl reading that recognizes the methylation. And they're taken off by the acetyl signals are removed by histone deacetylases, and methyl groups are removed by histone demethylases. So what we're going to do now is we're just going to talk about the principle of writing and erasing. So, and because it's easy to comprehend and understand a little better than methylation, which is slightly more subtle, we're going to talk about acetylation first. So if you think about wanting to unwind the genetic information, and you see the histones are not present here, they're removed, and this gene unwinds and it can be read by transcription machinery. How does that happen? Well, the lysines are positively charged residues, and DNA coils, or DNA itself, is a polyanion. Deoxyribonucleic acid is based on phosphoric acid, which would be highly ionized at physiological pHs. And a polyanion would unwind because the negative charges are repelling one another. So the histone charges help to bind the DNA into a compact form, and the histone tails are protruding from those complexes, those histone octamers. If the tails are acetylated at the Ks, this starts to allow the system to unwind. The histones dissociate from DNA. The DNA unwinds from a compact level to a level that can be read, and therefore the genetic information can be translated into RNA and transcribed into RNA and then translated into protein. Now, what's interesting is that lysine acetylation and deacetylation are in fact anti-cancer targets in their own right, because these can go wrong on certain genes. So those genes can be turned on and off by modifying the acetylation status. So we've got HATs putting the signals on and HDACs taking them off. This is just for information only, so you can see what's happening to the lysine tail. So this is the deacetylated lysine, and you can see that it reveals a positive charge because the amino side chain of lysine is protonated at physiological pH. A histone acetyltransferase transfers an acetyl group to the N-terminus, releasing CoA and putting an acetyl group on here, and the acetyl group has no charge, therefore it removes that positive charge. If the deacetylase is required, water and the enzyme action removes acetate. That goes into the system, which can tolerate acetate quite merrily, and the lysine is deacetylated. Excuse me. If a specific residue, again on a histone, is modified by acetylation, that's referred to as something like H3K-AC, which indicates acetylation, just in case you see this in your background reading when you're looking. Methylation signals on histones are a little more complicated. So we've got lysine in its deprotonated state. A single methylation can occur, a double methylation can occur, and a triple methylation can occur. And notice that in all cases the side chain is still positive. Now this is something that we're going to have to come to terms with, is that methylation status is different to acetylation status, and it's not so obvious how the methylation status modulates a gene on and off, but in fact it does, by a slightly different mechanism. So if lysine's been monomethylated on K9 on the H3 residue, it's referred to as H3K9ME1 if it's just monomethylation, and so on. Now remember, the methylation signals can be on multiple lysine residues on the histone tail, and these can be read by specific chromodomain possessing proteins involved in transcription. We're going to look at a little more closely at writing and erasing. So the methylation signals are written on the structure, on the histone tails by histone methyltransferases, and they're called histone methyltransferases because they transfer a methyl group from a cofactor similar to the acetylation in acetyl-CoA. It comes from a methyl cofactor containing cofactor, and that methyl is transferred onto the lysine side chain. The methyl tags, as we mentioned before, can be read by chromodomain containing proteins. So if we look, an HMT can put a methyl group on, or it can take a methyl group off. Or an HDM can take a methyl group off. And this can happen on each of these steps. Now what you'll notice here, just for a point of information, but you don't have to be too concerned about it, is that the demethylation of this group to here is actually performed by a different set of enzymes to the demethylases that are doing these steps, and in fact works by a totally different mechanism. So a histone demethylase removes a methyl group and reveals the lysine residue. And the mechanism is an oxidative demethylation mechanism. Now this is an important slide. If you notice here, the step of removing a methyl group, so this would be a demethylation, and so if we have two methyls here on a dimethyl, the same happens as here. It's an oxidative step. It occurs, it's mediated by an enzyme that uses FAD as a cofactor, and the oxidation occurs by removal of a hydrogen from the methyl group, and this generates something called an iminium ion, and the iminium ion is prone to chemical hydrolysis by water, and when that hydrolyzes by attack of water on this group, it forms a carbanolamine initially, and that carbanolamine falls apart to fall off as formaldehyde and releases the monomethyl lysine in this particular example. So let's have a look at the enzyme LSD1. So LSD1 is a histone demethylase, and it's quite an interesting looking protein, because I'm sure you're not used to seeing a globular protein extended in this way. So there's quite a long tail on the enzyme, and that tail interacts with another protein called co-rest. If we have a closer look at the active site, then we have FAD in the middle of the active site of the globular part of the protein, a cavity that can recognize the H3 side chain, and this is just a small run of peptides with appropriate groups on for crystallization. So this is an H3K4 peptide, just a small part of the protein, mimic, for crystallizing a structure for drug design and for understanding the mechanism of the enzyme. Excuse me. It's the enzymes are, and there's quite a few crystal structures of these enzymes with various drugs and various peptides binding to them. They nearly always have co-rest fragments within them, and that helps stabilize the crystal structure for recrystallization, or for crystallization I should say. The co-rest fragment is actually a protein fragment of the rest co-repressor. Now a co-repressor is a protein that helps repress genetic information, shut down a gene, so stop it being translated. The complex that contains our LSD1 is part of a family, and what we're going to do is just concentrate on this one here and not look at this one, which is why it's greyed out. So, and the reason for showing this picture is for two reasons. You can see that there's a complex of proteins, not just the enzyme itself, doing something to H3K4. What I wanted you to see is that LSD1's there with co-rest binding this, and that attracts other proteins which help recognize the genetic information, the gene. But we also have HDAC1 and HDAC2. These are two histone deacetylases, and so you can see that methylation and acetylation are actually very allied. They're closely working together, and this methylation of this K4 is happening in a bigger complex. LSD1 is part of several chromatin complexes, including a nucleosome remodeling. It's originally called the neuronal silencer co-rest, but it's also part of the NERD complex, which is shown in slightly greyed out colours there. So this complex is the thing that catalyzes methylation, demethylation of dimethyl lysine at H3K4. Now notice, so this is just for information only, but it's just to show you the latest thoughts on the structure. So this is a very, very recent publication that shows LSD1 in a slightly different orientation. You can see that the co-rest fragment here actually terminates in what's called a Sant2 domain, and remember, this is for only information, you don't need to know this, but you can see that this Sant2 domain actually binds to DNA, so that helps this complex recognize DNA. Then the blue chain also has an arm going off in a different direction to Sant1, which binds to HDAC here. And then another part of the LSD1 enzyme, which carries on from these enzymes are actually truncated, they're chopped short of what they actually are, and this one has a longer extension with a chromodomain on here. So one end recognizes DNA, the HDAC in the complex deacetylates DNA, changing its expression status. And then CHD1 here helps the enzyme recognize the methylation status of the histones within that gene, and then the enzyme does its normal job. This is and again for information only, this is a slightly closer look at how the methylation is recognized by a chromodomain. There happens to be a loop of amino acids that just sees the lysine with methyl groups on and allows a peptide to bind onto that domain.